my pleasure to introduce you to the chair and founder of the Minnesota Water Research Fund. Bernie Bullard has devoted the better part of his life to making sure that our water is safe. We are so proud to call him a University of Minnesota alumnus. Bernie has both his civil engineering and master's in business administration degrees from our university. And he is well known to many of you as a leader in the regional and national water and wastewater engineering field. Over his long career, Bernie has led St. Paul Regional Water Services, served as Director of Water Treatment and Distribution for the City of Minneapolis, and shared his talents with local firms such as TKDA, SL Serco, and CDM Smith. He's also been very active with professional associations such as AWWA, APWA, and many more. Bernie has incredible expertise. He is also incredibly passionate around ensuring that effective management of our water supply must be informed by sound research. He has a vision to have Minnesota and the University of Minnesota continue to be a leader in this area. And this is why he founded the Minnesota Water Research Fund through his own generosity in 2015. It's my pleasure to now turn things over to Bernie to tell you more about what inspired him to create the fund and what the committee is working on today. Thank you so much, Bernie. On to you.
It's great to have so many people joining us today for this webinar. This is the fourth research webinar hosted by the Minnesota Water Research Fund, sharing the efforts of our research fund with all of you. I spent my life and my career as an engineer focused on water. That's why I created the Water Research Fund here at my alma mater, the College of Science and Engineering in Minnesota University. You can see the Water Research Fund's vision and mission is on the screen. Water research has an essential role to play in the future of our cities and our state and our nation, and our lives and health in general. We cannot allow our knowledge, technologies, or water management practices to become outdated. Research is vital to ensuring access to clean water for the 21st century for all of us. The philanthropically supported Minnesota Water Research Fund currently supports faculty and student water research in the Department of Civil, Environmental, and Geoengineering here at CSE. Our vision is to enable the university to be a nationwide leader in innovative water research to improve public health and protect Minnesota water resources. We're already making an impact. In just 5 years, the Minnesota Water Research Fund has supported 6 research projects led by 7 CSE engineering faculty working with graduate and undergraduate students. We're getting ready now to fund 2 additional projects for this summer. In addition, our fund has invested in the purchase of water research equipment that will enhance research and teaching in CSE. We also regularly host these research webinars like this one to introduce a broader audience of alumni, water industry leaders, and external stakeholders to the water research happening here at our university. For more information on the Minnesota Water Research Fund, you can visit our website. At this time, I'd like to take a moment to thank and recognize the volunteer members of our committee, which is composed of university alumni and water and industry leaders whose names are listed on your screen there. Dave Allen, Lisa Cerny, Patricia Christensen, Bo Johnston, Andrew Ort, Naeem Qureshi, Michelle Stockness, Carl Street, and Stu Thornley. We meet regularly and they're an excellent committee. I'd like to now turn things over to the professor and head of the Department of Civil, Environmental, and Geoengineering, Joe LeBuzz, to get us set off in the next part of the program.
Thank you so much, Bernie. First, I'd like to welcome the participants to the Minnesota Water Research Fund presentation by one of my colleagues, Assistant Professor Judy Yang, and her PhD student, Shih-Hsun Huang. But before I introduce Dr. Yang, I would like to thank the Minnesota Water Research Fund under the leadership of Bernie Bullard for the continuing support of research conducted by faculty and students in our department. Bernie, thank you so much for your vision and leadership. Now it's my honor to introduce Dr. Judy Yang, who joined the faculty at the University of Minnesota in September 2020, 2.5 years ago. She received her master's and Ph.D. degrees in civil and environmental engineering from MIT, and she spent about 2 years as a postdoctoral research fellow at Princeton University. Her expertise is in the general area of sediment transport, but with a focus on the fundamental understandings of micron scale flow, particle, and bacteria interactions and their global impacts as identified by multi-scale experiments. And certainly you'll see some of that in today's presentation. Dr. Yang has already received several awards, and I'll mention only two: the Caltech Young Investigator Lecturer in Engineering and Applied Science, and the MIT Graduate Woman of Excellence. Besides being an outstanding researcher, Dr. Yang's teaching of our undergraduate fluid mechanics course has been noteworthy. She's a dedicated and caring instructor, and the student evaluations well reflect her teaching abilities. Judy, we look forward to your presentation, and I will provide the warm welcome. So thank you.
Thank you, Joe, for the great introduction, and thank you for the Minnesota Water Research Fund for funding our research. So my student Shih-Hsun will talk about the research he has been conducting, which was funded by the Water Research Fund. So now I just want to share a little bit of what's going on in my lab and the vision we have. And it's a great pleasure to be here. So the research in my lab is about multi-scale environmental transport. Specifically, we are interested in fluid sediment microbe interactions. So before I talk about the research in my lab, I would like to share with you two of the most important lessons I learned from COVID. First, microbes, these tiny organisms, do play an important role in global issues. And the second, not only the organisms themselves, but the interactions among the microbe, fluid, and the particles, especially their physical transport, matter. And the research theme of my lab is about fluid, particle, and microbe interactions, especially their physical transport, and the impacts of this transport on a wide range of environmental and health issues. There are 4 research topics that are based on similar fundamentals that we are interested in addressing. The first one is about coastal erosion. So each year, the United States lost about $8 billion due to erosion. And over half of the world's wetlands has lost in the past decade, in the past century. However, our prediction of coastal erosion and river erosion has been uncertain. And one of the major issues is we don't understand how the microbes, particle, sediment, and fluid interact with each other. Researchers show that locations with biofilms, the sediment transport rate, can alter by several orders of magnitude. But how these microscale interactions affect the macroscale behavior is currently unknown. And the second issue we are interested about is about soil carbon emission. Soil absorbs about 20% of fossil fuel emissions, and they play a critical role in global carbon cycle. And recent studies show that the majority of carbon emission from soil is controlled by the micron scale hotspots, which are those micron scale bacteria aggregates. So those activities control the carbon emission. But how those hotspots, which are this tiny scale, control the global carbon emission is unknown. And the third issue, which is very important for Minnesota, is harmful algal blooms. They are widely present in Minnesota's lakes. In the Great Lakes region, over $200 million have been lost per year due to harmful algal blooms. And one of the most promising ways to remove harmful algal bloom is to spray clay particles to water, which cause the algae cells to flocculate. But how does this micron scale sediment cell interaction affect the efficiency of this method, especially under flow condition, under windy days, is unknown. And the first topic we are interested in is about how bacteria spread in soil and lung. So we know soil has a lot of contamination, the incubator of pathogens. So how bacteria spread in soil is very important to understand to prevent a future pandemic. And in lungs, how bacteria spread in lung controls lung infection and is a cause of the exacerbation of many lung disease, such as chronic obstructive pulmonary disease, COPD, which is third leading cause of death in the United States. So as we can see, all those important environmental and health issues have something in common, which is they are controlled by the microscale fluid microparticle interaction or surface interactions. And those problems occurs across different scales. So the microscale interactions are impacting their macroscale behavior. Just for your reference, the diameter of our hair is 20 to 200 microns. So how can we understand how those tiny things are affecting this macro scale important environmental and health issues? So my lab is in a great position to tackle those important environmental and health issues because my lab have scales, have expertise across those different scales. And we have expertise in both physics and microbiology. So over the past decades, so we have conducted research at the large scale, conducting this flume experiment. So this is a 1 meter wide, 10 meter long flume. We have simulated rivers and wetlands in the lab and use this large scale facility to look at how vegetation generated turbulence can affect sediment transport. And our study has been highlighted in American Geophysical Union's newsletter as a paradigm shifting study. In addition to this macro scale expertise, we also developed the first 4D imaging technology of soil on a chip. So these are micron scale microfluidic channels that we fabricated in clean room. And we have used these channels to demonstrate how carbon are being sequestrated by single clay and how bacteria uses carbon in clay. And we provide suggestions on how to improve global carbon model prediction based on our micron scale study. In addition, we also looked at the mesoscale, how bacteria spread in a simulated soil. So in this study, we for the first time discovered that bacteria can spread in a synthetic soil by producing a flow along sharp corners. As you can see on this video, the bacteria self-generate a flow at a sharp corner. And we found out that this bacteria-generated corner flow is because bacteria can produce a chemical called surfactants that change the hydrophobicity of the surface. So this new bacteria spreading mechanism have a profound impact on our understanding of how bacteria spread in soil and in lung. So currently, my group is combining technology across these different scales to address the environmental and health questions I mentioned at the beginning. So we are currently combining microfluidics and flume experiments to look at how bacteria and clays affect coastal erosion and how carbon are being decomposed in wetlands. And we are also looking at how bacteria spread in soil and how clay can cause harmful algae cells to flocculate and sediment. In addition, we are also designing micron scale synthetic sediment and look at how biofilms are developing among sediment and glue sediment together, which affect sediment transport in coastal areas, also in pipes and in organs. So the goal of my lab is to combine technology across different scales to improve our fundamental understanding of fluid particle microbe interactions and their impacts on those important environmental and health issues, including coastal erosion, harmful algal blooms, soil carbon emission, and bacteria spreading. Currently, my lab is actively expanding, and I'm looking for motivated graduate students, undergraduates, and postdocs to join my team. So with this, I would like to hand the Zoom to my graduate Shih-Hsun, Shih-Hsun will introduce one small glimpse of what's going on in my lab.
Thank you, Professor Yang.
Okay, I'm going to stop sharing. Shih-Hsun, you can take over.
Okay. Thank you, Professor Yang. It's great to be here today. And today I will show you how we quantify the impacts of vegetations on hyporheic exchange in a laboratory experiment. So first of all, let's talk about hyporheic exchange. So in aquatic ecosystems, there was a region in the sediment bed where the flume in the sediments is actively exchanged with the flume of the surface water. And this zone we call it hypoic zone. Actually, this word is a combination of two words. The first one is hypo, means below. The second one is rios, means flow. So this is a zone that's beneath the surface flow. And in this zone, there was a hypoic exchange, which mixing the gases, water, nutrients, and even pollutants in the surface flow and subsurface region. So it controls the pollutants transport, biogeochemical cycles, and even the biodiversity in such aquatic ecosystems. So it is a very important process. And in the aquatic ecosystems, such as river, lake, wetlands, or intertidal zone, the vegetation is everywhere. We already know that vegetation has a great impact on surface flow and sediment transport. So we use vegetation as a process to restore the stream, to stabilize the riverbank, and increase the water quality. The problem is that we are not know much about how vegetation modifies the hypoic exchange in such environments. One of the main reasons is that vegetation and sediments are not transparent, so we cannot see through the sediments and know what exactly happens to the hypoic exchange when there was vegetation. In the previous studies, research used dye to illustrate the hypoic flow path, which is induced by the backbones of turbulence. However, these techniques can only map the flow path beneath the surface or along the sidewalls of laboratory flume. It cannot be used to study the hypoic exchange induced by vegetation, because this kind of exchange is happens around the vegetation and is a three-dimensional phenomenon. To overcome these difficulties, we innovatively use a transparent sediment called hydrogen beads. So in these two boxes, we can see there was hydrogen beads. And with these beads and a fluorescent dye, we can see through the sediment and observe the transfers of dye in the sediment, which can be a hypoic exchange process. So this is how we made hydrogen beads in our lab. The hydrogen beads is made of polysaccharides, and then we prepare polymer solutions and drop polymer solutions into the cation solutions below it, and then we can get discrete spherical beads like this. After we prepare all the hydrogen beads, we can set up our experiments. This is the flumes I used to simulate the river in our lab. This recirculating flumes is about 40 meters long and 6 centimeters wide. And in the middle of the flume, there was a string text section about 1.5 meters long. And in these text sections, like this, we fill the hydrogen beads in the bottoms of the flume as a sediment in our channel. And then we instill transparent styles to simulate the impacts of vegetation on the flow. I remember the purpose of our experiments is to quantify the hypoic exchange rate, which is induced by the vegetation. So the next step is we want to visualize the hypoic exchange process. What we do is we ingest the fluorescent dyes into the sediment bed. Because it is a fluorescent dye, so when we shine the blue lights on the dye will emit green light. What we need to do is to check the green lights, and then we can measure the transfers of dye within the sediment bed. This is the top view images of the test section. As you can see, the blue circles or blue dots are the dials we use to simulate the vegetation. And the black patterns are the meshes we use to hold the beads in place. And the green area is where the sediments mix with dye. When we conduct experiments, we run the flow in a channel which is from the bottom to the top. And you can see when the tides go by, the green lights emitted by the dye in the sediments gets dimmer and dimmer. And that is because the dye was leave the sediment bed. And each experiment, we can have a curve. That's green light intensity decrease over time. We call this curve washout curve. And there was 2 examples of washout curve. One is from the experiments without vegetation, and one is from the experiments with vegetation. In these figures, the x-axis represents time. The y-axis is the green light intensity, and the black line is for the case without vegetation. The red line is the case with vegetation. As you can see, the dye in the channel with vegetation is released out of the sediment bed much quicker than the dye in the channel without vegetation. Now we know that the vegetation increase the hyporic exchange rate. The next question is how much, because we want to quantify the impacts of vegetation. So we use a simple mass transfer model. In these models, all the parameters depends on the settings of the experiments, except one, the hyporic exchange velocity. So we can say that in these models, this hyporic exchange velocity determines how fast the dye release from the sediment bed. So if we fit our model to the curve we gain from the experiments, we can get a values of hyporic exchange velocity for each experiment, and then we can compare the results of different experiments. Here is the results we conducted experiments with vegetations and without vegetations under different mean flow velocity. So in these figures, the x-axis is the mean flow velocity of surface flow. The y-axis is the hyporic exchange velocity we just saw. And again, the black circles is for the cases without vegetation. The red squares is for the cases with vegetation. You can see that as similar flow velocity, the hyporic exchange velocity is much higher in the channel with vegetation. So we can say that the percent of vegetation increases hyporic exchange rate by a factor. And then specifically in our experiments, it's increased the hyporic exchange velocity by a factor of 4. And it also means that if we did not consider the impacts of vegetation on hyporic exchange rate, then we will underestimate it. To further predict the hyporic exchange rate or hyporic exchange velocities in the channel with vegetation, we propose a turbulent-based model. So in these figures, actually plots the same data set in the previous slide. The only difference is here. The x-axis now is the turbulent kinetic energy, which indicates how strong the turbulence is in a channel. And then we can find that the hyporic exchange velocity of cases without vegetation and cases with vegetation follows the same trend, which means we can use the turbulent kinetic energy to predict the hyporic exchange velocity. So here is the 2 conclusions. The first one is our experiments showed that the vegetation increased the hyporic exchange velocity by a factor of 4 as same mean flow velocity. And then we also show that we can use turbulent kinetic energy to predict the hyporheic exchange velocity in a channel with vegetation. We believe these experiments can advance our knowledge about the hyporheic exchange in a channel with vegetation. We also hope our results can help the future engineer to design their stream restoration projects, which includes vegetation. And finally, as mentioned by Professor Yang, they are the members in our research group. Thank you for them, and then thank you for listening. This is my presentation today.
There we go. Hopefully everyone can hear me. Thank you so much, Judy. Thank you, Shih-Hsun, for your presentations. My Zoom is being a little cranky today, so sorry for the delay. Just a reminder, go ahead and submit your questions in our Q&A feature at the bottom of your screen. We'd be happy to take those questions now. So we'll give you a minute while we're waiting for folks to submit their questions. I thought perhaps I could start the conversation by asking you, Shih-Hsun, if you wouldn't mind sharing. I think folks are always interested to know what inspired someone to pursue a PhD and to do research in this area. Would you be willing to talk for a few minutes about how you came along your journey to this point and kind of what inspires you to look into issues like the hyporheic exchange?
I think this is a good question, and I think I was always excited about new knowledge. And when I was young, I was like to read the new research results in the news. And I think maybe one day I can just provide my affairs on these areas to expose the new knowledge in the science. So that's why I pursued PhD degree.
Thank you. I also, and I don't know if Judy, if you want to take this, or Shih-Hsun, but I'm interested in the intersection of the modeling that you do experimentally with the flumes, as well as the modeling you do using computer science. Could you talk about how that interplay happens in your research group a little bit and how important, excuse me, both of those are to you?
Yeah, thanks, Shannon. We do a little bit of computer modeling or like theories. Most of our breakthrough results are from experiments. So yeah, so we found out it's important to expand, like to know what's missing in the simulation. Like a lot of hypotheses have been made in the simulation. So that actually inspires our experimental design, because we see, oh, this turn isn't unsure in this model. So we need to do experiments to learn more about the mechanism so we can implement better large scale models.
Thank you. I see a question coming in for Q&A, and I'll let either one of you decide who wants to answer this. So please, they asked if you could give a definition of the hyporheic exchange velocity.
Okay, maybe I can answer this question. So thank you for the questions. Because in our models, we have two control volumes and an interface. That's the tide will exchange between two control volumes. And we're using parameters called effective diffusion coefficient. And we divide these coefficients by the length of this mixing layer, and it will become, in the dimensions, this become the velocity. So we called it hyporheic exchange velocity. So that's how these parameters come from.
Thank you so much. I see a question coming in from Joseph, who says, is this model for hyporheic exchange in a stream or river system also applicable to near shore zones and estuaries? Does salt versus freshwater impact the fluid dynamics?
Yeah, maybe I add on. So just add on to Shih-Hsun's explanation of hyporheic exchange velocity. You can just think about like the surface water and subsurface water are constantly being exchanged, and the hyporheic velocity just give you a physical explanation of how fast these are being exchanged. So if it's exchanged faster, then the contaminants in the sediment may release to the surface water faster. And regarding the river or estuary, I think the physics is the same. So right now we focus on the physical transport, the fluid transport. We assume all the chemicals are following the water particles. But if they are salt, they maybe have some reactions like binding. So that may make it different. But our results about how vegetation affects this hyporheic exchange should be the same.
Thank you, Judy. I see a question coming in from Tanny who says, can you talk about the implication of this research for drinking water?
For drinking water, okay. I just, Shih-Hsun, you can add on. So for drinking water, actually many drinking water are coming from lakes or treatment wetlands or rivers. And I think our results show that if we have vegetation, we can actually use vegetation as a tool to increase the exchange between surface water, subsurface water, which may be able to help us use this mechanism to remove contaminants from surface water, actually help increase the water quality in rivers and streams, wetlands. So that may help us improve the quality of our drinking water, which are coming from surface water.
And I think in recent years, other than drinking water, recent years, it's more popular for using the artificial wasteland to do the water treatment. So I think our results can also help them to design such facilities. So that's another application other than the drinking water.
Thank you both. I've got a follow-up question, and I think you addressed a little bit of it, and this is also from Joseph. Is this model for hyporheic exchange in stream river systems also applicable? Oh, sorry, wait, there's one more. So if the hyporheic exchange velocity is faster with vegetation, does that mean that pollutants are released faster with vegetation?
That's a great question. So I think, so that actually go back to Shannon's question about like the modeling and the vegetation. So in our experiments, we show that the hyporheic exchange velocity is larger if we have vegetation, because vegetation generates this turbulence that makes it what's inside the sediment easier to release. And that's a condition that under the same flow velocity. So the flow velocity in the vegetation is the same. But in nature, we have patches of vegetation that the flow get diverted. So inside the vegetation, the flow velocity may be lower. So which is why we need to combine, actually, if we have a complicated system with vegetation patches, we have to combine simulation and the whole geography, the whole geomorphology to predict what is exactly the transfer rate inside the vegetation patch. But under the same mean flow velocity, yes, vegetation increase the release of contaminants from sediment. I hope this answers your questions.
I hope so too, Joseph. If you have additional questions, feel free to submit them in the Q&A. Bernie, I don't know if I see you there. I don't know if you had a question for Judy and Shih-Hsun. I want to give you an opportunity if you did. Not right now, okay. If you don't mind, I'd love to hear from you, Judy. You're a relatively new addition to our CG faculty, and we're so delighted that you're here. I wonder if you would be willing to talk for a minute or two about the process you go through to build your research group, to recruit the students who are going to work with you. What are some of the unique challenges that you face as you continue to expand your research group? Would you be willing to talk about that for a few minutes?
Yes, of course. Thanks for the question. I think the biggest challenge is actually the pandemic, because when I first arrived in the middle of pandemic, and so we have to, so all the purchasing and all the equipment arrived like half a year later. So we actually waited for half a year and we have to ask for permission to go back to the lab. But after the pandemic, things just get much faster. And yeah, and I'm very glad that Shih-Hsun is in my lab. Initially, I was a little bit concerned because Shih-Hsun has no like lab background, doesn't have any he didn't have any hands-on experience, but he learned. He's a fast learner. He like learned all the tools, how to do experiments, how to run flume, how to do imaging processing very quickly. And I'm very proud. And he made all the transparent sediment, transparent vegetation, so which make our lab very fun to work on. Yeah.
Thank you so much. Can you talk a little bit about the environment for funding for research in the water area? Obviously, you were able to get some seed funding from Minnesota Water Research Fund, and we're so happy for that. But can you talk a little bit about how you pursue additional sources of funding to keep your research going?
Yeah, sounds good. So first of all, we really appreciate the funding from the Water Research Fund, and my research, the research in my lab is more fundamental, like more fundamental mechanistic understanding. So we actually apply for NSF a lot and the Department of Energy. And recently we also start to work on some lung-related stuff. So NIH, National Institute of Health. And I think there are also some, I think Minnesota is a great place because there are also some state funds like LCCMR. So we also applied for the harmful algal bloom project. So I think being at University of Minnesota is a unique position to pursue this fund and to get more involved in the local water related issues.
Thank you. Thank you. Bernie, it looks like you've got a question. Go ahead.
Yes, when I was a student, I worked in St. Anthony Falls lab and then in the civil building in the wastewater lab, how I paid for most of my college. But is a lab research done out at St. Anthony Falls, or are you doing it right there in the civil building? I'm not really familiar with exactly where the labs are these days.
Yeah, thanks, Bernie. My lab is all at St. Anthony Falls laboratory. We don't have that much space in the civil engineering building. Yeah.
Okay, that's where I started. First job at the U was at St. Anthony Falls. So very good.
It's a great place to work.
It's a great resource for us to have on adjacent to our campus, not far away. Well, let me just check the Q&A to make sure I'm not missing. I don't see additional questions. So I think what I'd like to do is thank you both for your presentation. And I would like to turn things over to Bernie for a few wrap-up comments for today. Thanks to all of you for joining us. Bernie, would you like to go ahead?
Okay, I think I got unmuted. I want to thank Judy and Shih-Hsun for taking the time today to share their research with us. Many thanks to all of you for attending today's session and to our advisory committee for their time and leadership in the Minnesota Water Research Fund. We hope this will inspire you to stay connected with us at the Minnesota Water Research Fund. Thought partners are always welcome. If you have ideas for us, ideas for research, or ideas for the committee that things we need to tackle, tell us what's on your mind. What water research questions do you have? What areas of research would you like to hear more about in the future presentations? What types of research are needed? On the slide, you'll see how there are 3 easy ways to get connected to us. Posted right now, the mailing list and contacting myself, or I can get to anybody on the committee. If you have ideas or questions, reach out to me directly via my email or message me on LinkedIn. If you want to stay informed about our work or be invited to future presentations or events, you're invited to join our email list. Just contact Shannon and she will set you up. Advancing water research takes financial resources. In addition to funding research projects annually, the fund is up fundraising to create a chair in water research at the University of Minnesota. Every gift makes a difference. The online giving link is shown on the screen now, and if you have any questions, please don't hesitate to let me or contact Shannon. Thank you in advance for investing in our goal to ensure the University of Minnesota is a national leader in innovative water research that improves public health and protects and preserves Minnesota's water resources. Have a wonderful afternoon, everyone. See you next time. We're giving you a few minutes back here, so go out and enjoy the snow. Thank you.
Thank you so much. Take
Thank
care.
you.